Okay, our next talk is Vipin Sreedharan. Not bad. He's going to talk about Oqtans, and the subtitle is not on the title slide. Okay, I'll let you introduce it. Thank you.
So, hi, I'm Vipin. So, a PhD student from Gunnar Rash Lab, Memorial Sloan Kettering Cancer Center, New York. And I started working on Galaxy plus Oqtans since 2009. So, hope you have seen this slide from RNA-seq blog post stating that what is the immediate need for the RNA sequencing community. So what's really needed? So out of, I think there are different 6 different response came out of that, and 55 out of 143 response were like they need a more standardized data analysis pipelines. So with this, so I'll just introduce my talk title: Reproducible Quantitative Transcriptome Analysis with Oqtans. So this work is supported by Memorial Sloan Kettering Institute and University of Tübingen, Germany, plus Max Planck Institute in Tübingen. So basically, this is the general pipeline in RNA sequencing downstream analysis. So starting from read alignment and with colligative filtering of the alignment, some post-processing of read alignment, then transcript prediction or differential expression testing or transcript quantification, and then the final stage is like functional enrichment analysis. So with this, we already collected a few tools together and packed in a way that it's executable standalone way, and also it can be integrated into Galaxy framework. So the tools, the colored box which represents the above analysis step basically. So the one with the star symbol came from our lab, and the others basically we integrated into our system. Sorry. And they are we started integrating from 2011 onwards. So apart from this, the one which is very right in the slide, starting from MG, so they are right now available on our Galaxy instance, which is a public instance, and they will probably soon integrate into the Oqtans package. So MGene is, for example, MGene is an eukaryotic gene finding system, so using RNA sequencing data. So just want to give a brief history about the Oqtans development. So we started the service since beginning of 2009, but the Oqtans started December 2009 publicly. So these are the key events happened during the time of development. So the main one was in 2011 February we created the Amazon machine image with Oqtans tools. So but in that time we only included the tools from our lab plus DC package from EMBL. So after that, by end of 2012, we added a lot of the similar tools which can perform the same task also into the Oqtans package. So right now on our public Galaxy instance, we have 593 registered users, and I made a recent check for the last 6 months. For the last 6 months, I can see that just using the Oqtans tool, there are 336 active users on our instance, and 12 tools are involved in the Oqtans package were developed from our lab, and 15 from other open source community. So most of the tools, most of the RNA-seq tools were integrated with the help of the Galaxy development team. So with this, I'll just briefly introduce our tools basically. So first I will start with PalMapper. So PalMapper is a read aligner, read mapper program. So it accounts the variation awareness to the sequence reads, and also the speed. About the speed, it spits. I mean, if you use for the variation aware, then the speed seems to be like 1 million reads per hour on one core basically. And if you're not using the variation aware, then it will be like 2 million. So it's a bit slow compared to the faster aligner like Star. So the next step, basically the post-processing of alignment. So read alignment post-processing step can be done with the RNA-geek program. So which contains 3 different modules. So one of them called is simple alignment filtering. This is a SAS program, which basically look for the inaccuracies in the read alignment. Next step is the YAMT program. So this is a margin-based transcript reconstruction method. So which is using the RNA sequencing information, read alignment information to predict the transcript structure with the use of computational splice site predictions. So the differential testing step can be done with RDIF program, so which basically determines the difference in the experiments, how well they are differentially expressed. So if you look for the differentially expressed regions in the transcriptome, so and it implements 2 statistical tests, and if you have the genome annotation, then it will be so based on that you can select the statistical test. So for quantification step, we rely on ARC1 program, so which is which accurately determines the abundance of transcripts. With this, with this workflow, basically we have done many different experiments. So I just want to briefly go through one of this. This starts with the computing a fraction of differentially expressed or unexpressed family of genes from Arabidopsis thaliana genome, and for this we have got sequence from Columbia accession, that is the reference genome, plus the Canary Island accession. So basically, both of these genomes seems to be quite different, and yeah, we have used for this analysis. We basically used the Oqtans cloud instance, and so altogether within using XX large instance on Amazon, we could done this by 30 minutes, and it almost cost like $3.8. So with Oqtans, so if you look for the general steps, basically, so Oqtans provides a facility to to use different tools for the analysis step, basically. So this one can look for the accuracy of each program on the same data set, how they are performing on the same settings at least. So so for the last workflow, which or last work what we did based on from PalMapper to RGIC and then to DSeq to the GeneCenter program. So just showing the blue line. So Oqtans availability. So we have the public Galaxy instance running on our local data center, and there is a public Git repository, and we do have our own lab Git repository. So also there is a Amazon machine image available on AMI, Amazon AMI, and few. I would say most of our tools are in Galaxy Toolshed, and with all Galaxy wrappers and dependencies. And with this, I'm just summarizing. So Oqtans simplifies the RNA-seq analysis with many different tools, and also it's very easy to instantiate in a cloud platform or in an existing local Galaxy installation, and command line, of course, in command line interface too. So with this, so we are actively working on developing the technologies to advance the RNA sequencing data set. So I would like also, yeah, I would like to hear any experience from your side too.
Fantastic. Questions? Question?
Yeah.
Do you support multiple conditions? Like conditions like you have the state of, I don't know, let's say infection 24 hours.
Yeah, sure. So differential expression tests you can do with multiple replicators.
You said PalMapper is variant aware.
Yes.
So can you make use of known variants, like maybe you've done DNA sequencing?
Sorry.
Can you make use of, say you've done DNA sequencing and you know what variants are
Mm-hmm.
in your sample? Can you make use
So
of-
basically, you can provide the variant file. Basically, if you have a VCF file, you can provide that, and with that, it will compute the variation. So, I mean, with using variants, it's a bit slow compared to the other aligners. So we are working on indexing right now.
I'm sorry. According to your price calculation in Amazon Cloud,
Yeah.
the price, the cost was just for calculation for CPU use, or including the storage price?
No, it's not including the storage.
Oh, okay.
Yeah. Just a second.
I see. Thank you.
One quick question.
So do you have stuff? You just pointing me or something?
Yeah, I did.
Okay, here. I got it now. I do have stuff for comparing all the different aligners and calling methods. How do you approach that? It seems cool that you can sub
Yeah.
different
So.
methods in and out.
So basically in HITSEC meeting, so so my colleague is presenting a talk about the various alignment comparison methods. So but I don't have a slide actually. So
Sure.
I think we have done a better job with our parameter settings actually. So not with the default settings.
Gotcha.
So
And do you have, what do you use for the gold standard that you compare against? I don't know anything about RNA-seq, I was just curious.
So basically, what we did, like we just try to look for how accurately it looked for the spliced alignments.
I see.
So in that, so PalMapper seems to be performing better than Top Hat and Star. Also, we have a multi-mapper resolution program integrated into the aligner in such a way that it will fit the read into the right location rather than giving in mapping in multiple locations. So yeah.
Fantastic. Thanks, Vipin.